In this video we will see the synthesis of thyroid hormones. Thyroid hormone synthesis and secretion is a multi-step process, which begins with iodide uptake from blood and can be divided into 8 sequential steps as follows. Iodide uptake, apical secretion, oxidation, iodination, conjugation, endocytosis, proteolysis and secretion. Iodine is an essential raw material for thyroid hormone synthesis. Iodides present in the food is ingested orally. From there it is absorbed from the gastrointestinal tract into the blood. Supplementation of table salt means that the average dietary intake is approximately 500 micrograms per day. About 120 micrograms per day enter the thyroid at normal rates of thyroid hormone synthesis and secretion. 40 micrograms per day iodide diffuses back into the extracellular fluid and 80 micrograms per day is secreted by thyroid in the form of T3 and T4. Further 60 micrograms per day is secreted to ECF. Some thyroid hormone derivatives are secreted in the bile. Approximately 20 micrograms per day iodide is lost in the stool. The total amount of iodide entering the ECF is thus 500 plus 40 plus 60, or 600 micrograms per day. It means 20% of this iodide enters the thyroid, whereas 80% is excreted in the urine. All this is called iodine homeostasis. As we have seen that the follicles consist of many thyrocytes. The surface of the thyrocyte facing towards the capillary is called basolateral membrane. This side of membrane contains a special type of transporter called sodium iodide synporter. This channel transports 2 sodium and 1 iodide ion into the cell with each cycle. The iodide is pushed to the cell against the electrochemical gradient of iodide. This is called iodide uptake. The inner apical surface of thyrocyte, which is in contact with the colloid, has another type of transporter, called chloride-iodide exchanger or pendrin channel, through which iodide is secreted into the colloid and chloride is taken up by the thyrocyte. This is called apical secretion. The apical surface of thyrocyte has a membrane-bound enzyme which is called thyroid peroxidase or TPO. This enzyme oxidizes the iodide into the iodine. This step is called oxidation. The thyrocytes are typical protein-secreting glandular cells, which secretes a glycoprotein named as thyroglobulin. Each molecule of thyroglobulin contains about 70 tyrosine amino acids, and they are the major substrates that combine with iodine to form the thyroid hormones. The endoplasmic reticulum and Golgi apparatus secrete the thyroglobulin into the vesicles, which are then exocytosed to the colloid. As the thyroglobulin molecule is secreted through the apical cell membrane into the colloid, in presence of thyroid peroxidase, iodine binds with the tyrosine amino acids within the thyroglobulin molecule. This process is called iodination or organification. Tyrosine is first iodized to monoiodotyrosine 23% and then to diiodotyrosine 33%. Monoiodotyrosine and diiodotyrosine in presence of thyroid peroxidase couple to form triiodothyronine or T3 7%. 2-diiodotyrosine in presence of thyroid peroxidase couple to form tetraiodothyronine or T4 35%, also called thyroxine. This is called conjugation or coupling reaction. The thyroglobulin molecule, which is iodinated and conjugated, is endocytosed by the thyrocyte. Process is called endocytosis. The endocytosed vesicle containing colloid then fuses with a lysosome. Proteolytic enzymes cleave the iodine-containing molecules from thyroglobulin into T3, T4, MIT and DIT. This process is called proteolysis. Remaining molecules in colloid material are recycled. MIT and DIT are not secreted. These iodinated tyrosines are deiodinated by a microsomal enzyme iodotyrosine deiodinase. The iodine liberated by deiodination of MIT and DIT is reutilized in the gland. Finally, by the process of secretion, a T3 and T4 are secreted from the thyrocyte into the blood. This is all about the thyroid hormone synthesis and secretion.